Thank you very much, and thank you, Paul, for inviting me and for your gracious welcome here to Europe. As I've been talking to people here at the conference, one of the things that seems to come up a lot is just how sick our patients are becoming. It used to be that on a rare occasion you'd see someone in a wheelchair walk into your office, but it seems more and more that our patients are sicker and they're younger. So we have to be prepared for trying to understand why they're sick and how we can really impact them. I have 2 lectures today. The first part is going to be a general overview of an approach, and it's using a systems biology approach to the practice of clinical care of very, very sick patients. When I was in medical school, one of my professors, one of my mentors, Dr. Lewis Barnett, taught me that you have to know what kind of lens you have on your camera. Some people have a zoom lens and they want to do a specialty and that's all that they want to do, and some people have a wide angle lens. And I was planning to go into specialty until I realized, no, I really do have a wide angle lens, and so that's the approach I'm hoping to bring to you today. So systems biology says that it's not enough to understand only one part of a system when studying the complexity of biology. There is in Seattle, Washington, an Institute for Systems Biology. It's a nonprofit organization that was started in the year 2000. And they look at systems biology and they're trying to understand how whole networks work in order to cause complex problems. And this is a quote from the Institute of Systems Biology, that systems biology is a holistic approach to deciphering the complexity of biological systems that starts from an understanding that the networks that form the whole of a living organism are more important than the sums of their parts. This is an example of a network of networks, which is really what we're dealing with when we're dealing with human beings. So at the very bottom level here, you're at a subcellular level of genetics, and then you work your way up until you get to organ networks, and then I would go even one further and talk about whole social systems and whole human networks. So today what I'm going to try to introduce is the systems biology approach to studying chronic illness. And we're going to be looking at genetics, at external triggers, at something that a topic that I'm introducing called the somatobiome, and then what the body does to respond to those different aspects. So when we're looking at genetics, we're really looking at multiple different things. Most people talk about genetics, they're thinking about SNPs, what is a person's individual mutations or variants, genetic variants. But the field of genetics is really much more broad than that. We have genomics, which is the complexity of how one gene affects the other. And certainly we had some discussion of that with the discussions on epigenetics. We have transcriptomics. Transcriptomics is looking at the RNA. So once a gene is transcribed to RNA, it's looking at a broad range of all the RNA that's being produced at a particular moment in time. And it tells you what genes are being upregulated and what genes are being downregulated at that moment in time. This really may be the wave of the future for human medicine, as we're looking at what's going on now. We do a treatment intervention, and then we can see those changes and see improvements to prove that our treatments are having a beneficial effect. Proteomics is the study of the proteins that are produced. And again, it's not looking at what is this one protein, but what are the interactions of many different proteins. So these studies really require the use of massive computer systems and people far smarter than I am to figure out how all of these things interact. And finally, there is metabolomics. And metabolomics is looking at the metabolites of these proteins, and often that is done through urine. And again, it's getting a sense of what is being up and down regulated in a particular individual. When we're looking at the genetic template, there's something that a friend of mine, Dr. Bernstein, coined as the home biome, and that is the sum of all the genetics that are at play in an individual. And it's not just what you're born with, but as we all know, what our environment has done to our genetics, what genes are being up or down regulated because of methylation. There's also an interesting feature of genetics in that just because you have a gene doesn't mean that it's expressed certainly in every cell. I mean, we all know that. But some gene mutations are uniquely expressed in only certain tissue types. So as we dig into genetics, we start realizing that it's far more complex than just looking at someone's SNPs and saying, oh, they have a COMT, that means X. It's far, far more complex. So when we're looking now at the external sites of triggers, what is triggering our body to respond, there are several areas that we'll be looking at. In the skin, you have infective organisms, so bacteria, fungi, viruses that infect our skin. Topical products that we use affect our bodies. Trauma, physical trauma, skin breakdown, and I don't know how well studied it is in Europe. In the US, we're just beginning to have some acceptance and understanding of how electromagnetic frequencies can affect people. And again, that is penetrating through the skin. The GI tract is certainly, as we've heard from both Dr. DeRid and Paul, a huge area for the introduction of triggers. There are allergens, there are infections, there are biotoxins, non-food substances, chemicals that we consume. There's a lot of use of glyphosate in the US. It's difficult to find any bread that doesn't have glyphosate unless it's organic. You all are far more advanced here than we are. But phthalates, styrenes, a whole slew of things that we consume, our bodies have to respond to. And then we also, because we use the enterohepatic circulation to try to detoxify, that can also be another source of triggers and toxins, is if we're reabsorbing those toxins. And finally, as Paul discussed and Barbara discussed earlier, the tight junctions and intestinal permeability. This is a slide from Dr. Alessio Fasano. If you don't know his name, he's probably the premier researcher studying tight junctions in the US. He's currently at Harvard. And this is a slide showing the tight junction here, and when it breaks down, how food particles on this side of the lumen enter into the body. And this is a more complex slide that shows the same thing. You see here, in this case, there's gluten attaching to receptor causing the production of zonulin. About 2/3 of the population carries at least one gene of prehaptoglobin 2 that produces zonulin. 1/3 of the population only makes prehaptoglobin 1, so they're less at risk of making zonulin. But zonulin comes out, it causes the destruction of the zona occludens protein of the tight junction, so this whole tight junction complex breaks open, and then food particles, bacteria, LPS, all sorts of different external factors can enter in to the body. And here we have the immune system poised and ready to protect us. In the respiratory tract, we have many other routes of triggers, certainly oropharynx, the alveoli. The olfactory nerve is tremendously important, especially if you have patients who have multiple chemical sensitivities. This is a route through the airway that toxins can enter in and people can respond. We just heard about the importance of the hippocampus and a lot of the limbic system structures. The olfactory bulb is right there. So when we inhale volatile organic compounds, they go up to the olfactory bulb and we get a reaction. Water damaged buildings, and this is going to be the big part of my second lecture, are also a tremendous source of inflammagens. And it's not that we consume mold. Mold doesn't really enter us as significantly through the gut. It really is through inhalation. Pollutants are also important, and as I mentioned, VOCs. Psychosocial triggers cannot be underestimated. I'm certain you all have patients who are very sick and have been very sick, and if you dig, you will hear stories of traumas, of childhood events, abuse, physical as well as emotional. You don't have to personally experience a trauma, but witnessing a trauma can also impact, be an important psychosocial trigger. And then something that we call ACE, or adverse childhood events. What we experience as children, the traumas we experience, follow us through the rest of our lives unless we actively try to break that cycle. So the microbiome, as we heard earlier from Dr. Marzetti, is the study of microbial communities that live in and on our bodies, and the role that they play in human health and disease. And when we talk about the microbiome, generally we're talking about nasal passages, oral cavities, skin, GI tract, and the urogenital tract. The problem with the term microbiome, as far as I'm concerned, is that when we use it, we generally mean gut flora. So there's the scientific research, which microbiota is the bacteria and the genes, and their genes, and that's our microbiome. But really common usage is that it's our gut. So I'm proposing the use of a new term called the somatobiome. We've heard a lot, Dr. Brascano spoke a lot about bacteria in our bodies, Borrelia, Babesia, Bartonella, etc. And we ignore those when we talk about the microbiome, and yet they're still within us. So the somatobiome I'm proposing is a collection of all the living organisms that are present across all the various ecological niches of the body. And one that is, I think, largely ignored is the vascular biome. What's going on in your bloodstream? I don't have on there the connective tissue biome. But what are the bacteria that are in our bodies in all their various forms? And so you can look at it from a couple of different categories. There are beneficial bacteria, those that help us make vitamin K in our guts. There are commensal bacteria that may just be hanging on for the ride. We're great big taxis for bacteria as we walk through our worlds. And then there are infectious. Types of organisms, well, we all know those from microbiology. So we have our genetics, we have these external triggers, and then the somatobiome, and then we have our body's response. Two people can both be exposed to strep throat, and one person will have a sore throat for a few days and low-grade fever and be fine, and the next person can have a very high fever, thigh pain, sore throat pain, and be sick for a week. Same bacteria, totally different presentation. And we all see this daily in our practices. So when we're looking at why someone is sick, some of it is their genetics, some of it is external factors and the somatobiome, but how their body responds, that's where their symptoms lie. So there are two ways of looking at the body's response. One is the cell danger response, and many of the slides that I'm presenting here are taken from Dr. Naviaux's work. I have a quite extensive bibliography at the end of my slide section, and I should note there's also a sort of how-to manual to follow through with what I'm presenting today, as well as some other information. So the cell danger response is evolutionarily conserved metabolic response that protects cells and hosts from harm. They're triggered by chemical and physical or biological threats that exceed cellular capacity for homeostasis. When the cell danger response persists abnormally, the whole body metabolism and gut microbiome are disturbed. The collective performance of multiple organ systems is impaired, behavior is changed, and chronic disease results. And I think we tend to underscore or under-focus on the fact that behavior has changed, and that's what we see every day. The patients who walk into our practice and they are sick, and that's what they know, is they know their sick behaviors. They know that they feel that they want to stay home, they don't want to go out, they don't want to go to work. So this is a little bit more detail about the cell danger response. We have what are called, we experience what are called PAMPs and DAMPs. And PAMPs are pathogen-associated molecular patterns. They're exogenous proteins that follow a certain pattern that induce a response. And DAMPs are danger-associated molecular patterns, and they're endogenous signals from unhealthy cells. They're known as the hidden self. And they're often from the mitochondria. So we'll be talking a little bit more later about the mitochondria, but mitochondria are thought to have originally started as bacteria that entered into cells. And so some of their internal mitochondrial components are very similar to bacteria. So they're really ideally poised to be able to be recognized by our pattern recognition receptors when a cell is in danger, when a cell has been disturbed and those contents start to come out. There are many different kinds of pattern recognition receptors. The ones we hear a lot about are the toll receptors. Toll receptors are found on the cell membrane, and they induce mitochondrial reactive oxygen species. Nod-like receptors are in the cytosol, and both nod-like receptors and toll-like receptors recognize both PAMPs and DAMPs. C-type lectin receptors recognize carbohydrate structures on pathogens, and the Rig-like receptors are also cytosolic, and they're found in both immune and non-immune cells, and they're very important for our immune response to viruses. They produce they cause the production of a lot of pro-inflammatory cytokines. So in addition to the cell response, we have a whole systemic danger response, and I like to think about it sort of in 4 different categories. First is our immune response. We heard a lot about that from Paul just now. But we also have our autonomic nervous system response. It triggers the hypothalamic, pituitary, adrenal axis, and then even within the mitochondria, we have our responses. So in immunity, what we see is T helper cell responses. I think Paul did a lovely job describing Th1 and Th2 immunity as well as Th17 autoimmunity. The innate immune response really isn't very elegant. It's a slam it down as quickly as you can, a lot of cytokine production, whereas the acquired immune response is where we make antibodies, and that relies on antigen presentation. So if you don't have adequate antigen presentation, you're going to have a difficult time kicking in that acquired immune response. Mast cells are a special kind of immune cell that I'm actually finding quite a bit of a problem in my patient population. They live in connective tissue. They release mediators. Histamine is the most well-known mediator. And when it comes to mold, not only are mast cells likely activated, but we see a reduction in the breakdown of histamine, and we see increase in histamine receptors. So we actually see more of the mast cell effect, even if mast cells are only slightly increased. And then within the nervous system, microglial cells are the immune cells that Dr. Dered talked about yesterday. The autonomic nervous system, I understand it used to be called the vegetative nervous system here, really is made up of several different components. The sympathetic or fight or flight response, the parasympathetic, which is restore and repair, and then let's not, of course, forget about the enteric nervous system. If someone has a spinal cord injury, they may need help breathing. They may not be able to walk or move their muscles, but as long as you access their stomach with a gastrostomy tube, their intestinal tract works because the enteric nervous system is separate. It is unidirectional. There's a study that took piece of intestine in an animal still attached. They cut at both ends and tried to reverse it, and the food didn't flow. So the GI tract is very, very intelligent, has its own nervous system, and we can't just think of it as just another organ, the GI tract. Other areas of importance really are vagal tone. Stephen Porges has written a book called The Polyvagal Theory that looks at some of the evolutionary development of the vagal nerve, that we have initially the vagal nerve caused us to freeze. I don't know if you have an animal like an opossum, that it plays dead when it's under danger. And then we developed more the fight or flight response. And then in humans, we've developed a social response. So we read social cues. And again, going back to that systems biology and the behavior part, that's an important piece to this. If vagal tone is off, it's going to be very difficult for someone to get completely well. And finally, the limbic system, from the hippocampus on, is an important part of the danger response. The hypothalamic pituitary axis, we've heard a lot about this today. The hypothalamus produces corticotropin releasing factor. I don't know of any way of measuring that. The pituitary releases adrenocorticotropin hormone, and we can measure that. I'm going to suggest that we look at both ACTH if it's available to you, as well as cortisol, so you see how they're communicating. And then the adrenals make various hormones. Mitochondria are important in a danger response because that's where we get our energy. And so if you're in danger, you need to be able to fight or run away. You need the energy for that. We see increased blood flow to muscles, to brain, to vision, so you can think, so you can see better. ATP increases. ATP is our energy source. And remember, ATP can act as a damp. So the more danger you're in, sometimes that can be a problem. Affected organs with a mitochondrial cell danger response, the brain, muscles, and the autonomic nervous system. Cyclical amplification is, I think, what we tend to see in our patients. They're sick. Due to an external factor, it causes an internal response, which makes them more susceptible to external factors, which makes them more internally sick. And we can see that in a couple of different areas. Dr. Lambert yesterday talked about the cell cyclical amplification in the immune system. Someone gets an infection, it suppresses their immunity. Stress also suppresses their immunity. So then they get a new infection, which further impairs their immune system, so they get another infection, and it can go on and on. We see the same kind of cyclical amplification in mitochondria. Mitochondrial dysfunction leads to autonomic dysfunction. Remember, the autonomic nervous system always has to be on, so it has a high number of mitochondria, high ATP requirement. And when you have dysfunctional autonomic nervous system, then you get decreased cardiac output, you get poor blood flow, you get hypoxia in tissues, so the mitochondria see less oxygen, so you get more reactive oxygen species and more mitophagia, more loss of mitochondria. The limbic system also can have cyclical amplifications where emotional traumas affect the limbic system, and that causes more sympathetic overactivation, which causes more emotional trauma. So how do we unravel this? We have to look at the individual response. Really, patients come to us with symptoms. They don't come to us saying, well, my enteric nervous system isn't working well today, please fix it. So we have to look at their symptoms and try to understand what systems are being impacted. And then so this is our job is to identify the pathology. We want to search the patient's history for exposures, and we're looking at these environmental triggers that may be causing that danger response. I put on here dental history, and I want to acknowledge the dentists who are here today, because dental pathology is really, really important. We'll talk a bit later about infections, but heavy metals, inflammation really can start in the mouth. And then our job is to test. We can do direct tests, we can culture, we can look at toxin levels, we can look at indirect tests, look at measures of inflammation, inflammatory markers, antibody responses, and then we can test externally. We can test buildings for water damage. We can test the insect if you've been bitten by a tick and you still have that tick. You can test it for organisms. Treatment priorities. In my mind, I think we have to improve the immune system. If your immune system is suppressed, it's really difficult to eradicate organisms. And so you want to focus on the things that are going to be causing the immune system suppression, and then you treat the infections, and then you really try to go after that cycle resetting from the cyclical amplification. So, do we have time? Okay, so I'm going to keep going for a little bit more. Today I'm going to be focusing on taking that systems biology approach and applying it to the exposure to water damage buildings or mold. You may have heard this term as Chronic Inflammatory Response Syndrome. You may have heard it called mold illness. We're now trying to call it environmentally acquired inflammation, specifically water damaged buildings, but there are other forms of environmentally acquired inflammation. But today we're just going to talk about water damaged buildings. So there are lots of different symptoms. In your handouts, there's a list of a review of systems that goes through all sorts of symptoms that patients may have, and you can have them mark how severe their symptoms are. And then there are additional symptoms that weren't included in that initial review of systems. There are lots of biomarkers you can look at. These can be expensive. They may or may not be available in your country. So some of this can be useful, but it may not be, depending on where you are. So the first thing you have to do is suspect that someone may have a water damage exposure and that may be contributing to their illness. And what are some things that may cause you to suspect that? Well, do they have Lyme disease that just isn't responding to treatment? Are they overweight and cannot lose weight? Do they have low testosterone? If you have a woman who's postmenopausal and you're trying to start BHRT, is she not responding to that? You're having a difficult time managing their hormones. Treatment failures, chronic inflammation. You can read this list, but some of the things that I think are more important: memory problems, thirst, frequent urination, chronic fatigue, cough, shortness of breath. So in order to be an environmentally acquired inflammation detective, you have to suspect a problem. You have to see evidence of biotoxin exposure, symptoms and signs. You have to diagnose it. You have to understand the tests that are available, and do a physical exam, and then hopefully treat and see improvement with treatment. The external triggers in environmentally acquired inflammation due to water-damaged buildings are not just mold. It's a toxic stew. Just as Dr. Borascano talked about the dirty needle that are ticks, our buildings can be really toxic stews if they've been water damaged. One thing I have noticed being here in Europe is that some of the older buildings here are most likely colonized with molds that are less toxic than some of the newer buildings. So I've had plenty of patients who say to me, oh, it can't be mold, my home's only 3 years old. But they have a very tightly sealed home that's gotten water intrusion, and mold can grow very quickly. And it's these newer molds that tend to be more toxic. But it's not just molds. There are bacteria, mycobacteria, Actinomycetes, VOCs. This is a long list of the biotoxins and bioinflammagens that can cause problems. So I'm going to show you some photos. This is mold, probably from after Hurricane Katrina, and you can imagine that pretty much anyone will be sick if they try to go live in this space. This is what some people might call mild mold, and say, oh, I wouldn't be sick there. But for certain people, they might be very, very sick if they lived in this space. This is mold behind wallpaper. Wallpaper glue seems to be a preferred food source for a type of mold called Stachybotrys chartarum. So the black mold that you see there is also, we in the US tend to call it black mold, is Stachybotrys. Here's mold growing under a sink. So don't just look on the walls, look under your sinks, under the plumbing. This is a hotel in Cary, North Carolina, close to where I live, and has a lovely water feature in the middle of the atrium. And it looks lovely until you look closer and see all the mold up on the ducting. I was there for a conference. I said something to the staff. Went back a year later, it was worse. I went up, I said, I was here last year, and the woman said, yeah, we remember. This is an interesting picture for a couple of reasons. First of all, you can see the water line. This was flooded, and they just simply moved all these things back in. Here's a box with water damage. It's crazy. But the reason I have this slide is because of this. Do you see this whitish, greenish powder on that surface? That's probably an Aspergillus species mold. There's nothing special about this photo. You can take any cell phone with a flash on, snap a picture, and if you see that, you'll see it with your naked eye as well, but if you see that whitish green, you know that that's mold. So it's something that's very easy for patients to do. This is condensation on the inside of a restaurant. It's a restaurant that bakes its own bread for their Subways sandwiches. Or sub-sandwiches, I guess I gave away the restaurant. And so this is on the inside. If you're in a building that has condensation on the inside, it is too damp. You're going to get mold growth. So in order to grow mold, what do you need? You need spores, it's the seeds. You need the right temperature, and where we live is the right temperature. You need the right nutrient source, and what we build with and live with is the right nutrient source, and you need moisture. So the one thing that you can change and you can control is the dampness of a space. This is a diagram of a building and envelope and all the various places where you can see mold and water intrusion. So here you can have plumbing leaks, you can have intrusion through windows and doors, through roof leaks. You can have up here in the bathroom or even in the kitchen, if there isn't adequate ventilation and you take a nice steamy shower, that moisture is going to condense on the walls and you can run the risk of having mold growth. And then this illustrates what's called a stack effect. So if you have moisture in your basement, in the US we have a lot of what are called crawl spaces, so the homes are built off the ground. So there's no true basement, but there's a space, generally about yay big. There's dirt, and then the house, and moisture can really build there. And the stack effect is like a chimney stack. The moisture will travel up into the house. And the statistics are that during the winter time, when the heating is on, about a third of the air that you breathe is coming from below, from your basement or your crawl space. According to the National Institute of Occupational Safety and Health of the U.S. and the EPA, about a quarter to half of all U.S. buildings have water damage after the hurricane that we just had and the one that's coming. It's probably going to be higher. Acceptable humidity really depends on the temperature outdoors. So if it's very, very hot outside, you want the humidity inside to be about 55 to 60% or less. If it's very cold outside, you want it to be much lower. So you will see online 30 to 60% humidity is acceptable, but if it's -5 Celsius outside, you do not want to have 60% humidity. It's too humid and you can have mold growth. These are what I call the Fab 5. There are 5 molds that are measured in a test called the Hertzsme test, and they're important because they represent different moisture requirements. So the first 2, the Aspergillus species, don't require as much moisture. They're more likely to grow in an HVAC system, in the ducting, the heating system. Chitomium and Stachybotrys, the 3rd and 4th, require a lot more moisture. So they're going to be growing if there's a plumbing leak, if there's a roof leak, if you have someone spill water every day, if the bathtub overflows and it's not dried up. Wallemia is typically found outdoors, and that also indicates that there's intrusion from the outside. This is a photo of Stachybotrys. Has very sticky spores, and so you're not going to see a lot aerosolized. The numbers required to have a problem with Stachybotrys are much lower than they are of the Aspergillus species. It requires very, very high humidity. And this is Aspergillus. It's very common in damp homes. The photo of the sandwich shop that had moisture on the inside, they probably had Aspergillus growth there. And so what is the risk? Well, the risk is really from inhalation of these airborne particles. There's some direct toxic effect as well as chronic inflammation. And a big problem is asthma. There's a wonderful study out of Scotland that showed that the #1 predictor of asthma severity was the Hertzsprung score, the amount of mold in their home. There's another study out of China that looked at rates of asthma in children based on the mold content of the schools, and the schools with the highest mold content had the highest rates of asthma. Yeah, I have one, just a little bit more. These are just 2 quick slides of symptoms. I won't read through this. There are lots of symptoms that can be associated. It really is not it's a multi-system, multi-symptom illness, and so there's a lot of symptoms that can occur. A lot of them are neurological and psychological and cognitive. So I think will be a good time to take a break. When we come back, I'll talk more about what we can do as clinicians to diagnose and then what we can do to treat. So thank you.
So we have a break now for 20 minutes, and then afterwards, Dr. Rapaport will continue her lecture, and you can ask questions afterwards. Thank you.